So today I will be talking about the FDA's perspective on the physiologically based pharmacokinetic analysis for biopharmaceutics application. And as you all know that this field is an advancing field, and there are multiple terms used to describe PBPK analysis for biopharmaceutics application. Throughout my talk, and in the literature, one of the terms being used is physiologically based biopharmaceutics modeling, PBPM. It's much easier to say it. So throughout my talk, I might use these terms interchangeably. Just wanted to bring to your attention. So next slide, please. So I will briefly touch upon the biopharmaceutics applications supported by PBPK analysis at the beginning of my talk. Then I will give you the brief overview of the regulatory landscape of the drug applications that contain PBPM data submitted to the FDA. I will also talk about the general considerations for model development and evaluation in supporting drug development. I will go over what type of information that should be in the PBPK study report for biopharmaceutics applications when the sponsors or applicants submitted to FDA. I will present a case study highlighting all the topics I covered during my presentation, and I will conclude my talk. Next slide, please. So the purpose of PBPM is, as we all know, that to link the in vitro dissolution or other in vitro testing inputs to in vivo exposure via PBPK modeling. And here are the few examples of categories of applications. As you heard, that, for example, it is very useful in doing formulation development or establish clinically relevant drug product specifications for conducting quality risk assessment and also drug product life cycle management. This approach potentially reduces the number of in vivo BA/BE studies. Next slide, please. As you know that FDA issued a draft guidance for the use of PBPK analysis for biopharmaceutics applications in October of 2020. And I provided the link here. You may access it in this link. So to this date, a total of 36 PBPM submissions submitted to FDA, which includes the investigational new drugs, INDs, NDAs, ANDAs, supplements, NDA and ANDA supplements for FDA's review. And of these 36 applications, 32 of them submitted in the last 5 years. And now the guidance is issued that I expect this number to increase. So there are two pie charts here I'm showing, shows the current regulatory landscape of these drug applications which contain PBPM data. On the left, of all applications, the purpose of the majority of the submissions were to set clinically relevant drugs product specifications, which were such as CMAs, critical material attributes. This might include particle size, specs, polymorphic form, or CPPs, and followed by dissolution specifications. And third category is biowaiver request, and lastly, to assess formulation-related impact, such as impact of protein pump inhibitors on drug exposure. Here shown on the right, the majority of applications belong to class 4 and 2 drugs, followed by class 1 and class 3 products. Next slide, please. So I'm providing here the general workflow of PBPM models. I will go over each box in the next slides, but briefly, what we like to see is the general strategy in development of PBPM models should include model objective, model development, and validation. And if it's all found acceptable, model application. Next slide, please. Here we see the examples of model objective. One example is development of a clinically relevant dissolution specification, which includes dissolution method and dissolution acceptance criteria or criterion. The ideal goal is to develop a biopredictive dissolution profile, which can be used as a surrogate method for in vivo performance. We have seen the PBPM is also used to widen the acceptance criterion or criteria at the FDA. And secondly, other clinically relevant drug product specifications, for example, setting the particle size spec or solid dosage form, can be established based on modeling predictions, which to ensure bioequivalence of batches with the specification limits set based on the modeling is bioequivalent to the clinical batch. There is a case study which I will cover later in the talk is on the example of setting the particle size specification. We also know that during drug development, there could be CMC changes such as formulation, manufacturing process, and site changes. Bridging between the clinical batches and to be marketed commercial batch is needed, and modeling will be able to aid to support that bridging. PBPM data could also be used to support biowaiver requests for post-approval changes. It is important to note that the model objective should be clearly specified in the submission. Next slide. So the data needed for model development and verification could be categorized in 3 layers here. The drug data, which includes drug substance physicochemical properties, formulation attributes such as particle size, solid state form, dissolution, and ADME properties of the drug product. Systems data includes anatomical structure and physiological parameters for GI tract, for example, gastric emptying, gastric volume, regional permeability differences, or another organ tissue whenever applicable. Clinical data includes clinical study design, dosing regimen, study subject population of the in vivo studies selected for model development and verification. Next slide, please. Model validation. Actually, for model development and verification, all the assumptions you have made should be also clearly specified. I forgot to mention that. For model validation, the predictive performance of a model should be validated for its intended purposes. That's really important. The validation acceptance criteria should be established a priori. Independent data sets not used in model development should be used in model validation of the model. And next slide. So virtual BE studies are used to predict the PK parameters based on the in vitro data representing the desired limit. For example, representing desired limit of particle size, which is the case study. The sponsor should consider the following when conducting the virtual BE studies. The number of subjects in virtual BE studies should be estimated based on the specific intra-subject variability using power analysis, and the number of virtual BE trials used to estimate the probability of conducting BE should be justified. And as you see, the conventional BE limits are acceptable. However, if this data fall outside the BE limits, additional data such as exposure response data can be used. Next slide. When submitting a PBPK study report for biopharmaceutics applications for FDA's review and feedback, the following information should be included. A modeling summary report elaborating the modeling strategy, the objective of the model, the modeling flowchart covering the model development, verification, and validation, rationale and supportive information on model parameters, formulation and in vitro dissolution information, and clinical study design, and the number of subjects, in vivo concentration profile data, and with this non-BE data also preferred. For virtual BE trials, description of intra and inter-subject variability, and as I mentioned before, justification of number of subjects and trials used in the BE studies. And if you are planning to submit a PBPM report for FDA's or Division of Biopharmaceuticks review, early interaction with the agency is highly encouraged. Next slide, please. Here's the case study. The objective of this case study of this model in this case study was to support the selection of API particle size distribution. API was the BCS class 4 weekly basic compound for immediate release drug product. Model development was conducted with experimental data are used for API-related input parameters. Default physiology parameters were used. IV data and additional clinical studies are used in model development. However, model validation was done with another set of clinical data which were not part of the model development. Then, model application was to set the right final drug substance particle size specification. And this was selected based on the parameter sensitivity analysis and the virtual bioequivalence study. Slide 12. Here in this slide, summarizing what I just said, but in a workflow. This workflow indicates that drug PhysChem properties, IV data, oral PK data, particle size inputs were used in the model development, and what model is validated by other set of oral PK data with different particle size distributions and with different doses. Model application was to support the particle size spec based on the virtual BE study results. Next one. So here we see on the top that failed virtual BE results when particle size spec was outside the specification. And on the bottom, when the virtual BE passes, then the particle size is within the PSD spec. In conclusion, next slide. So in conclusion, the use of physiologically based pharmacokinetic analysis for biopharmaceutics applications establishes a link between in vitro and in vivo. It provides a regulatory flexibility, potentially reduces the number of in vivo BAPE studies. FDA supports innovative, data-driven, mechanistic modeling approaches, and early interaction with the FDA regards of using modeling approaches is highly encouraged. And with this, I conclude my talk, and I'd like to acknowledge my colleagues in the Division of Biopharmaceutics at the FDA. And if you have any questions, I'm happy to answer.
Well, thank you very much, Banu Zolnik, for that very nice summary of the FDA perspectives on PBPK. One question that kind of comes to mind is that a lot of times with these PBPK models where you're incorporating physicochemical properties within your modeling, some of those are easily to be attained, some of them more difficult for larger molecules. From a, as you're reviewing these submissions, do you give more weight of importance to if some of those physical chemical properties are derived experimentally or, estimated through in silico methods?
I guess this question depends on where you are in the development, but if it's coming to us, if there is something, let's say solubility, right? It is better to use an experimental data than the predicted data. We would ask that type of question because you should have experimental data at that point.
Okay, excellent, makes sense. As these types of drug molecules are being developed, I'm sure you're seeing more and more PBPK models being submitted, and along with the the first guidance about how to structure the report, and then the one you mentioned in October of last year for kind of the conduct. Is there any preferences within the FDA of one platform versus another for developing a PBPK model?
One platform meaning software, or
one software platform, or one approach, building it within like a non-MIM structure versus a GastroPlus versus a SimSip.
No, we are all open to different innovative approaches, as long as it is validated and verified, developed well and validated. So we do not have any preference, any platform.
We've got some nice questions coming in through the chat. One of which is, how does the FDA view cross-validation for the model validation step?
So the internal validation, you mean percent PE, or what's the right criteria?
I believe that may be what they're getting at, or whether you're using cross-validation, like between different molecules, perhaps? How critical that is versus validating.
Well, I think, let's say within a given, we don't have the set percent of, let's say, PE error right now. However, again, that becomes the objective of model and available data and application of the model. It is all basically risk-based approach on those parameters. We don't have the right, at this point, the cutoff rate yet.
So that
the more
seems
we
like,
learn about this.
Go ahead, sorry.
Right, the more we learn about it, and the more we would know what that value should be.
Which I think is more evidence of, communicating with the agency earlier is always more beneficial.
Absolutely.
I've got a question of, do sponsors and regulators ever disagree on whether or not a model should be considered validated? And if so, how are such disagreements resolved typically?
So if there's a disagreement about the validation for a drug application, obviously, we might not be, that model at that point might not be applicable for that model objective until we get more data. We will try to find other ways to set the particle, like let's say set the spec or other applications. But if your model is not accepted by the FDA, we will provide a lengthy deficiency comment And then this doesn't mean we are not going to see your model ever again. We might need more information, and then you could submit the requested information, and then we will review, and then we can approve the model that way. So it is ongoing communication, basically.
Along the same lines of gathering more data, the data that's used in the PBPK model or PBPM model building, does it all have to be under GLP conditions, or is non-GLP data also accepted in building those models? The person specified in vitro, but I think I would expand that to both in vitro and any preclinical data you might have available.
Right. I think these data, the ones we see, is under always GMP.
How often do you see in vitro precipitation studies and precipitation models in some of the submissions you've been getting in the last few years?
Yeah, we are, depending on the drug molecule, if there is a potential for precipitation, let's say later in the GI tract, yeah, we do see those as well.
Okay, excellent. We've got a few more moments if anyone else has any questions. And I don't see any coming through. So again, I think we can leave this one early unless there's any last questions and move to the next session. Again, I remind all the attendees that as we close out this particular speaker session, to leave and then rejoin next with Liang Zhao. Thank you very much, everybody. Thank you, Banu Zolnik.
Thank you.